Hey guys, welcome to my channel. This is Omar the Great. If you're new to this channel, remember to subscribe. Please hit that button below. Hit the subscribe button below. And don't forget, tap the bell button so you can always get notifications of my upcoming videos. So this is going to be a series of highly requested videos. People have been asking me about excitable tissue. So the series that I'll be shooting will be typically excitable tissue physiology. So what are excitable tissues? When we talk about excitable tissues, So these are tissues that have the ability to generate and propagate an electrical chemical impulse. So these tissues are of different types. We have what we call the nerves, okay, or nerve tissue, which can be excitable, okay, or nerve stroke neurons. I will tell you the differences. We also have muscles, okay. These muscles can be cardiac, okay. They can also be smooth muscles, and they can also be skeletal muscles. So globally, these excitable tissues, we're going to be looking at each of them in details, okay. And today we shall start with the nerve tissue or the neurons. But all these excitable tissues have some properties in common. One, they are, just from the name, excitable, so implying that they have a properties. So let's look at the properties. So under some of the properties, these tissues are actually excitable, okay. They are excitable. So what does excitability of a tissue mean? So excitability, it means that this tissue is able to respond to a given stimulus that comes to it. So excitability is the ability of a tissue to respond to a stimulus. Okay, that is one. Two, there is what we call conductivity. So an excitable tissue also should have a property of conductivity. So this means, we talked about in our definition, this tissue should be able to propagate, okay? They should be able to propagate or transmit the impulse. So this is conductivity, the ability to conduct or propagate or transmit a given impulse. Another thing is these tissues, because we said we have some muscles here, is what we call contractility. So this is another important property of these excitable tissues. So contractility is usually attached to more of these muscles. So the ability of these muscles to shorten or to reduce in length following changes in tension that apply to them, okay. So they can contract, you reduce in length, or you change in tension, as we shall see. So these are most important properties of these muscles, properties of these excitable tissues in general: excitability, conductivity, and contractility. All right, then let's look at our neuron. So when you talk of neurons, what is a neuron? A neuron, this is the basic structural and functional unit of the nervous system. So if it is a structural and functional unit of the nervous system, then what's about the nerve? A nerve, the difference between a neuron and a nerve is that a neuron is found within the central nervous system, whereas the nerve, this is the ones that are found within the peripheral nervous system. Remember, the central nervous system typically is made of the brain and spinal cord, simply, okay. So these neurons and the nerves are structurally similar, okay. They have just a few peculiar differences. So these nerves can actually be we can classify these nerves into different classifications, okay, or neurons. So, for example, the first classification here they can be classified according to the direction in which they are transmitting their information. So the direction of information. So according to the direction of information, this can be, you could have what we call the sensory nerves, okay, or neurons, and the sensory nerves, okay. We have those that are sensory stroke afferent, and then we have those that are efferent or motor. motor stroke efferent. So the difference between these the direction. So the sensory or afferent are taking information to the central nervous system, whereas the efferent are bringing information away from the central nervous system. The second classification can be according to which structures they engage in, which structures are they mostly found. Here we can find this according to the structural location. We can have what we call the visceral, and then we have what we call the somatic. So the visceral are found more in the visceral tissues. Okay, these are especially within different visual tissues like the GIT, you know, or different organs. Whereas the somatic, these are found in non-visceral structures. For example, you can have maybe the skin, the muscles. Okay, so another classification. This can be classified depending on the number of processes that stream out from the cell body. Okay, so according to the number of processes, we could have what you call differentiate them into three categories. We have what you call unipolar, you can have bipolar, and then you could have multipolar. So this means that the number of processes that stream out from the cell body, we are going to look at the structure. So if you look at the structure, we shall exploit it. So you find if this is the cell body, so when you have only one process streaming out from the cell body, that is a unipolar. Okay, when you have two structures that are streaming out from the cell body, okay, two processes that are streaming out from the cell body, that becomes a bipolar. When you have three structures that are streaming out, more than two, so you have several structures that are streaming out, different processes streaming out from the cell body, then this is what we call a multipolar. The other classification can depend on whether they have myelin or no myelin. So you could have what we call the myelinated and the unmyelinated neurons or nerves. Myelinated, and then we have the unmyelinated neurons or nerves. So this is the global classification of these neurons or nerves, as we are going to see. So after us knowing the different classifications of these neurons, I think we need now to dwell more in our structure, the structural arrangement. So when you look at the structure of a neuron, we said we need to know that we can divide it into two things. One, primarily it has what we call a cell body, okay, or what we also call the soma, but also is also called the perikaryon. Perikaryon. Okay. The other thing is after us knowing that this is the cell body, we have the cell body. The other process, the other structural arrangement is what we call the processes. So these processes can be what we call, you can have an axon and then you can have a dendrite. So these processes, let's look at the structure here. So you see from our structure, this is the cell body or the soma. So typically the whole of this structure here is what we call the soma, okay? The soma, okay? But this cell body is structurally a cell, okay? So it has all the most of the cellular components. So if you missed our video on cell physiology, you can review this about the cell. It will tell you more of the details. So importantly, some organelles that are put here is what we call a mitochondrion. You have of course the cytosol, cytoplasm, and then the nucleus, okay? And then this the Golgi apparatus, as we shall see, it packages so many vesicles after synthesizing some neurotransmitters. It has some other important structures here which play a big role in protein synthesis. These are called nisobodies. And these nisobodies have protein synthesizing machinery, Okay, so they contain a lot of ribosomes that play a role in protein synthesis. So these usually do, as we already said, and then send information through the endoplasmic reticulum to the Golgi apparatus that does a lot of packaging. So, for example, in this typically is the neurotransmitters. Okay, so it processes and releases, produces these neurotransmitters, which are packaged in form of vesicles and actually can be transported. So that is the cell body. Then the processes, we have the dendrites. So if you look at these processes here that are attached to the cell body, these are called dendrites. They are a little smaller, and also the synapse, they're the receptive pole. The receptive pole, they play a role to receive a lot of information, okay, that is coming within the cell. So the receptive pole is typically made of the dendrites and also the what? The cell body. Whereas the axon plays a fundamental role in transmission. It plays the transmission. So it is a transmission pole. The transmission pole is the axon. Okay. So stretching from here to here is the axon. So this to here, this is the axon. So this axon, it usually starts from a point called the axon hillock. And then just after the axon hillock, you have an initial segment. We are going to see the function of this initial segment when we start talking about the electrical changes that occur and the transmission of this impulse that will be occurring along the neuron. And then we have this, you see these structures here, these ones. This is actually myelin. This is a myelin sheath. A myelin sheath. Okay. So let me tell you something about this. So these myelin sheaths are produced or synthesized from cells that are called Schwann cells. And these cells are found, the Schwann cells synthesize myelin within the peripheral nervous system. Within the central nervous system, the myelin sheath is produced or synthesized by oligodendrocytes. Okay, so usually if there is a problem with the oligodendrocytes and they're not producing enough myelin sheath, then there is going to be a problem with the conduction of an impulse, because myelin play two roles. They have an insulatory role and also play another role in conduction of the impulse or propagation. It's typically what we call the cytotary conduction. And so these are also called nodes of Ranvier. We shall see more of the differences when we talk about conduction. So if there is a problem in the synthesis of this myelin within the central nervous system, that's maybe especially by the oligodendrocytes. So that is maybe there's an autoimmune process that results in the production of antibodies against these myelin sheath producing cells. This results into a condition called multiple sclerosis. So multiple sclerosis is a very big condition. So these patients get extensive weakness, especially of their muscles, and a little slack because they have difficulty in propagating or conducting these impulses. Then the other structure here is what we call the presynaptic knob, also called, you see some books telling you Bolton, and others also called the terminal. So this is the global structure of our neuron. So you see this, let's explore more about the axon. The axon plays a role in something called axonal transport. So there's something in the neuron, there's something in the axon called axonal transport. Or what you call axoplasmic flow, so irrespective. So I've magnified here our structure of the axon. So the axon plays a very big role in establishing a communication between the cell body and the terminal Bolton or the presynaptic knob. And so this connection is a connection of movement. So implying that the axon can transport molecules from one side. This is usually called on the cell, this is usually the minus end of the minus end of the neuron, and this is the positive end. So usually transport molecules from the minus end to the positive end of the neuron. And so there are two forms of transport that we can look at. Two forms of axonal transport. There's what we call the anterograde axonal transport, and also what we call the retrograde axonal transport. So this typically is the anterograde, and this will be the retrograde. So anterograde is moving from the minus end to the positive end, and then the retrograde is moving from the positive end to the minus end, back to the cell body. So there are differences in this. Anterograde is classified into two. We can have what we call fast anterograde and slow anterograde. Okay, so fast anterograde, this difference in the speed. This one anterograde, the fast anterograde moves molecules at around 400 millimeters per day, whereas the slow moves molecules from around 0.2 to about 8 millimeters per day, whereas the retrograde will move molecules at a speed of around 200 to 300 millimeters per day. So this fast anterograde transport or axonal movement helps to move molecules, for example, especially vesicles. Vesicles, as you can see here, these vesicles are typically vesicles, so it does a lot of vesicular transport. And these vesicles contain things like the neurotransmitters. Okay, so these neurotransmitters are usually packaged in vesicles, and they are transported across or along this axon. And so they are important proteins that play a very big role in this anterograde axonal transport. And these proteins are here. You see this helical arrangement of these proteins. These proteins are called kinesin proteins. So these kinesin proteins are typically of a class called the microtubule associated proteins, or what we call the MAP proteins, microtubule associated proteins. So these are different classes or categories, okay? And so because they are structurally arranged within the microtubules. And then the other one that plays a role in retrograde transport, this is called dynein. And its structure arrangement, it's usually associated with another molecule called dynactin. And so this one is typically a membrane microtubule associated protein. It's a MAP1C, MAP1C type of protein. So these are also in different families. So there are different families, and for them they are usually called the KIF family. So you can find KIF family 1, 2, 3, and the rest. So these move molecules. So they usually bind these neurotransmitters and move these molecules. The movement of these molecules is usually energy dependent. So each of these molecules, the kinesin and this dynein, they actually have intrinsic ATPase activity, which has the ability to cleave ATP, to cleave these phosphate bonds to release energy. So this movement is actually energy dependent. So that is an important thing to remember. So usually moves neurotransmitters, but also can move what we call the mitochondria. Okay. One thing I didn't tell you is that the axon, the axoplasm, actually has less of few or even none of protein synthesizing machinery. For example, those niso bodies and the ribosomes and the prokaryotic are absent within the axoplasm. And so the axoplasm constantly needs a constant refreshment or rejuvenation of these new proteins that it's using. Okay, maybe it's using, you see the enzymes that may take part here. We have different proteins, and your propagation of any as we shall see. So these need constant rejuvenation or refreshment. So there is need to keep receiving some molecules or synthesized protein that are within. Okay, and these come along with the fast anterograde transport. And then the slow anterograde transport. Usually these microfilaments, when they get old or they get denatured or damaged, they need to be replaced or repaired, the microtubules and microfilaments within. So these will move by the by the slow anterograde transport. So this move much of the cytoskeletal structures, okay, the slow anterograde transport. Whereas the retrograde transport, this one moves molecules, usually degraded proteins. So you can say degraded proteins. Usually degraded proteins are taken back by this mode of transport and taken back to the lysosomes within the cell body, broken down, denatured, and then the proteins and peptides and amino acids are reused for synthesis of other proteins, new proteins. So besides having the degraded proteins that it transports, this actually can also bind some other molecules from the presynaptic knob, and then they are transmitted back into the cell body. Some of those molecules include what we call growth factors. There are some growth factors which are also called neutrophins. Okay, so these growth factors can include what we call the nerve growth factor. We can also have a brain-derived growth factor. So there are different growth factors for the nerves, okay, that play very important roles, because nerves that are within the peripheral nervous system can have an ability to regenerate, okay? And so that's why sometimes in when there is damage to the nerves, maybe someone had an accident and they have some problems with nerve conduction, especially within the peripheral nervous system structures like the lower limbs and the rest. So if the origin is from that area, so if they undergo with time under physiotherapy and with a lot of support, they can actually have some nerve regeneration over time. Whereas if you get damage in the central nervous system, you may actually have no regeneration. And some of the other reasons, one is that in the peripheral nervous system, these neurons can actually generate some growth factors that can be utilized to stimulate this regeneration of growth. But also two, the myelin sheath in the central nervous system, which is produced by the oligodendrocytes, is a little different from the myelin sheath produced by the Schwann cells of the peripheral nervous system. That in the peripheral nervous system, the outermost layer coating of those cells has something called a neurolemma, and this neurolemma has cells that can actually undergo some mitotic division to regenerate these nerve cells. So that is a structural difference between a property of a difference between these neurons within and the nerves. Okay. Then, besides moving these growth factors, the other thing they can actually move is some viruses. Retrograde transport can actually move some viruses. And this, for example, we have what we call the tetanus toxin, okay? And also you can move what we call the rabies virus, but also can move herpes simplex virus. So some of these viruses, they have their receptors here, okay? For example, they're typically tyrosine receptor kinases. This is a family of receptors, TRK receptors. These usually bind these molecules. It binds this molecule, usually, for example, and this superfamily of these tyrosine receptor kinases can actually bind these growth factors. So, but irrespective of the molecule, when they bind, they have specific receptors that bind on this terminal bottom membrane, and then they are endocytosed. Okay, endocytosis. The membrane undergoes an invagination to close off the receptor, and this is actually forms an endosome. Okay, and this endosome is what is transported by this dynein protein, as we talked about. Okay, and then back to turns to the lysosome, and then it undergoes the processing required. So some of these, for example, if you look at you get bitten by a dog and on your foot, and you're bitten by a dog on your foot, but over time you're going to get neurosymptoms that are originating from the central nervous system. So they use this, they optimize this retrograde mode of transport to actually spread those viruses, to spread this virus infection. Herpes simplex, also rabies, tetanus is also they utilize the same processes. So yes, this brings us to the conclusion about our neuron. So what we shall talk about in our next video is going to be, we are building from here. We shall talk about much of the postsynaptic knob, the presynaptic knob, and how it functions, and the physiology of the neuromuscular junction. Thank you guys for listening. Thank you so much for watching. Please don't forget to subscribe and hit that bell button so that you can always receive notifications of my upcoming videos. We meet next time. Thank you.